USDA RS in Florida, and he's gonna present the research that we have been doing since 2020 and 2021 on anaerobic soil disinfestation, looking at alternative carbon sources. Thank you, Raymond.
Thank you, Francesco, and thank you everyone for being here. Yeah, so the title of the presentation that I'm going to be offering to you all today is evaluating byproducts of the agri-food industry as carbon sources for anaerobic soil disinfestation application in Pennsylvania high tunnel vegetable production. So a little background on high tunnels in Pennsylvania. They're becoming more and more common, and the reason behind that is that they have the potential to extend the growing season and provide more control to the grower over the environment that the crop is exposed to, and also protect the crops from harsh environment, overall, usually resulting in better crops like higher productivity and higher quality. In terms of management, the high tunnels in Pennsylvania are managed intensively and sometimes even year-round, which can lead to nutrient buildup, and also with the limited crop rotation that is available for farmers that have high tunnels, and the microenvironmental conditions that are characteristic of high tunnels, like higher humidity and higher temperatures on average throughout the year, can lead to decreased soil health and increased incidence of soil-borne pests and pathogens. And even though this is an emerging issue, considered like an issue that is relatively new, we're already seeing this. This is a tomato plant that grew in a high tunnel here in Pennsylvania. We can see two fungal pathogens and one nematode, one pest, nematode pest. Well, we can encounter bacteria, we can encounter viruses, and like a plethora of different disease, like soil-borne disease agents stemming from poor soil health. So as a farmer, if you still want to produce, if you still want to have a high tunnel that's productive, what will you do? Since the second half of the 20th century, the most preferred approach was chemicals, like synthetic chemical application through soil disinfestation. And among those chemicals, the most widely used was methyl bromide. But methyl bromide in the beginning of the 90s was deemed an environmental pollutant because it was an ozone depletant. So up to the 90s until the 2005, it was phased out. There were some other like critical use exemptions that some industries were able to pull off using them up until 2016, but up to this point, most of the industry in the US does not have access to methyl bromide. So if you're a farmer today that has a high tunnel with disease issues stemming from poor soil health, what are your options? So many farmers are using resistant varieties from breeding programs or shifting to grafted tomatoes using resistant rootstocks. And also some farmers are steaming their soils. Soil steaming has seen some technological developments that farmers have been able to employ soil steaming without having so many like expenditures in terms of fuel and time. But still, in Pennsylvania, there's still a need for a viable non-synthetic soil-borne pest and pathogen management strategy, and that's where anaerobic soil disinfestation can help. But what is anaerobic soil disinfestation? It's an approach to manage soil-borne pests and pathogens that was originated at the beginning of this century in the Netherlands and in Japan simultaneously. And ever since, it has been researched in different parts of the world, including the US, particularly in the US, in California and Florida. It's a biological method of controlling soil-borne pests and pathogen populations. So it relies on the native microbial population for soil-borne disease suppression. It is applied in 3 steps. The first one is to amend the soil with a source of easily available carbon. The second one, tarping the soil with totally impermeable film to prevent gaseous exchange with the atmosphere. And the third step is to irrigate the soil until saturation. So in this way, the soil pores are mostly saturated with water, so the oxygen is displaced. The tarping of the soil prevents gaseous exchange with the atmosphere. And while those conditions are on the soil, carbon source is being decomposed. So as the carbon source decomposes, the oxygen is depleted. So there's an initial growth in microbials and microbial communities that stems from the amendment of the carbon source that is like by aerobic decomposition that then shifts to like mildly anaerobic conditions in which facultative anaerobes can keep decomposing the carbon source and then obligates anaerobes keep decomposing the carbon source. So there's a shift in microbial community that goes hand in hand with anaerobic conditions developing. And in that process, the carbon source is still being decomposed, and the products of anaerobic decomposition of this fermentation process is tied to pest and pathogen suppression. So microbial community, anaerobic conditions, and alternative products of respiration. These are the factors that sum up to the mechanisms and the effectivity of anaerobic soil disinfestation and pathogen suppression. So carbon sources, the carbon source is a critical aspect of anaerobic soil disinfestation, especially in a place where it's newly introduced, because there's no ideal carbon source for all the contexts. And in a place where it's newly introduced, like in Pennsylvania, we have to look at what's available, what's out there in the market, and what is the potential for farmers to get it. And that leads us to the objectives of our study, which are to optimize the application of ASD for Pennsylvania high tunnel vegetable production by looking at the byproducts of the agri-food industry and seeing which one of these byproducts of the agri-food industry can elicit good anaerobic conditions under the relatively low temperatures that characterize Pennsylvania, because we have a relatively short growing season compared to other states where anaerobic soil disinfestation has been applied before. And then after we apply the carbon sources in ASD, we want to evaluate what are the phytotoxicity effects, potential phytotoxicity effects, and the nutrient dynamics after the application of the carbon source. For that, we identify byproducts of the agri-food industry, and we characterize them by their total carbon content, nitrogen content, and total mineral content. With these values, we define application rates and use them as carbon sources for ASD. The ones that we chose were spent grain from the production of beer, mushroom substrate from the production of mushrooms. Here is a big industry in Pennsylvania. Wheat middling, which is a byproduct of wheat flour production and wheat bran. It has less value than either of those, and it's usually just used as cattle feed or livestock feed. And then edamame that gets produced, like byproducts of edamame get produced in the packaging of edamame products. These 4 byproducts of the local Pennsylvania agri-food industry were to be compared with molasses, which is a standard carbon source for ASD that has been tested in different parts of the US and the world. So there were 2 application rates. There were 2 application rates for each of the carbon sources. If we look to the left, all the carbon sources were balanced by the total applied carbon with a target rate of 5,167 pounds per acre of carbon. And then the same carbon sources were applied also at a half rate of the ones balanced by carbon, and they were supplemented with poultry manure to achieve a total applied nitrogen of 268 pounds per acre of nitrogen. And that way, manipulating the carbon to nitrogen ratio of all of the carbon sources. So all of this treatment is considered an augmented factorial in split plot in randomized complete plot design. So this square here, that molasses, is a split plot in RCBD, but because we included the untreated controls, the whole treatment becomes an augmented factorial. And there were 4 replications. Every time that we see PM from now on will be poultry manure. Are there any questions up to that point? Okay, so we're not in the results section yet, but since we use the values of the total applied carbon and total applied nitrogen per carbon source to determine the application rates, I wanted to show these first here. So the application rates of all the carbon sources in terms of like the total organic matter that was applied of each carbon source are summarized here, the percent of carbon, percent of nitrogen, and then Each carbon source that was applied at full rate without poultry manure, you'll see the 5,167, and each carbon source that was applied at a half rate supplemented with poultry manure, we'll see the 268 applied nitrogen in pounds per acre. So each one that has the supplementation of the carbon source with the poultry manure, you'll see that it has a lower carbon to nitrogen ratio. And we wanted to observe, like it has a lower carbon to nitrogen ratio compared to when it's like the carbon source by itself at a full rate. And we wanted to see how this would translate into anaerobic conditions and the microbial change. We were using a movable high tunnel. This was at Rock Springs 2 years ago, September 14. That's when we started the ASD process, the ASD application. And we were using a movable high tunnel that would be rolled on top of these beds once the ASD incubation period was done. Carbon sources were incorporated at a depth of approximately 20 centimeters. And then this is the totally impermeable film. And underneath, there would be irrigation lines that we would use to apply 5 centimeters of water. So the soil parameters that we monitored throughout this incubation period were the redox potential, which is a measure of anaerobicity in the soil, pH, electrical conductivity, nitrate availability, and the temperature. 56 days after that application, we planted Solanova green lettuce at 2 rows per bed, 25 centimeters in row spacing, and you see that the high tunnel is already on top of the beds. They grew as a winter crop for 135 days, and as you can see from this picture, there's no signs of phytotoxicity. They were harvested last year on March 24th. And to observe differences in development in relation to the treatment that they were planted on, we did a biometric assessment for fresh weight leaf area, and once we dried them, we calculated some other values: dry matter content, specific leaf area, leaf weight fraction, and leaf area ratio. So once the lettuce crop was harvested, it was removed. We removed the high tunnel from on top, and we planted red Deuce tomato in last year, May 25, and they would grow as open field tomatoes on those very same beds that we had planted the lettuce first. We continued monitoring nutrient dynamics through soil sampling, and because we wanted to observe what was the residual fertility, like we mentioned before, like what were the nutrient dynamics in the soil after the application of the carbon source, we applied no fertilizer whatsoever. So these tomatoes, no fertilizer whatsoever. And we harvested 6 times. You can see that the tomatoes, this is extra large tomatoes, it's a big quantity here. We had a lot of tomatoes. And this picture on the left bottom, you can see the grading system that we use more or less. We were using the USDA measurements. Any tomato that was extra large was 7 centimeters in diameter or above. Tomatoes that were large were 6.3 to 7 centimeters, medium 5.5 to 6.3, and anything that was below 5.5 was considered small. We also took measurements of the pH, the juice inside the tomato pH, Brix, which is a measure of sugar content, and acidity. And after each harvest, we took a representative sample, dried it, ground it, and sent it for analysis for total nutrient availability. Okay, so now we're officially, well, sorry about that. It's so the x-axis here. Sorry about that. Well, I'm going to explain the x-axis then. The x-axis is September, October, November, December, January. So this period here is months, and then it also shows the days after treatment. It stems from September 2020 up to July 2021. So I think we can get the picture more or less. What I wanted to show here is the average air temperature, because ASD is a biological process. We need to think about temperatures here in Pennsylvania, especially because it's an issue because it's colder than in other areas of the US. And we can compare the air temperature to the soil temperature. The x-axis is the same. Soil temperatures do not reach 0, and it's probably because we put the high tunnel on top. and that prevents us from reaching freezing temperatures. And then the dashed blue line is the experimentally found temperature that ASD can be applied. That's the minimum temperature that ASD can be applied, around 15 to 16 degrees Celsius. So we had a good window here, and this is when we applied the ASD for ASD application. So this is the soil redox potential time course graph. On the left, we have millivolts per 1,000, so 300,000, 200,000, and then this is hours, the hours that the treatment went on. 200 is the threshold for anaerobicity. Anything below positive 200,000 millivolts is considered anaerobic. And we can see that the treatments of wheat middling and wheat middling supplemented with poultry manure reached the lowest point of anaerobicity. The critical period for the redox potential to go down and have a biggest effect in the production of anaerobic products that are suppressive for pests and pathogens, and the biggest microbial shift is in those first 500 hours, more or less, which is around 21 days. So this is the biggest shift. And you can see that all of the treatments became heavily anaerobic, with middling being the highest or the lowest. These results of anaerobicity with the redox potential can be better observed with our calculation of cumulative redox potential, which can be obtained with the values of pH of the soil and that graph that we just saw. And on those first 21 days, with middling, molasses, and edamame are the highest, but you can see that from the error bars that they're not significantly different from each other. We had a lot of variation. And the controls were also anaerobic, but that's because, well, we discussed that that's because we also irrigated those first 5 centimeters of water. They also received that, so that caused anaerobic conditions. 56 days after, the results of the cumulative redox potential were not very different from each other. And the soil pH, as the anaerobic conditions developed, the soil pH went down. We believe that it's because of the production of soil organic acids, from the decomposition of the carbon source. But as the treatment went on, and the year passed, the pH came back up. And that's the general trend. For EC, the electrical conductivity, it went up towards the beginning. It was kind of erratic. It's difficult to tell a pattern there. But towards the end of the season, of the tomato planting cycle, it came back down. And it's a similar pattern with the nitrate, in which a lot of nitrate was being produced in most treatments. And then by the end, probably because of the heavy feeding of tomato, it came back down. Okay, so the way that we approach the statistical analysis for this part is that we were looking at factor A, which is the carbon source type, factor B, carbon source rate, and then we were looking separately at the untreated control and the untreated control plus the poultry manure. In the first part, we're going to be talking about the lettuce results. The lettuce results show no differences in leaf area, fresh weight, or dry weight. And then the macronutrients that they were able to obtain throughout the season in terms of the percent in their tissues. No macronutrients were observed to be significantly different from each other. But here we report sodium. Sodium is not a macronutrient, but it's more a worry in terms, like, not so much of a deficiency, more a toxicity. So we need to report it, especially for growers that want to employ anaerobic soil disinfestation, that the higher rate of carbon application can result in a higher uptake of sodium. And then the accumulation in the lettuce plants in terms of milligrams per plant, we were just talking about milligrams per kg, but milligrams per plant, we see that the actual difference is very small, like 1 milligram per plant, although it's significant. Then looking at the micronutrients, none of the micronutrients seem to be significant except for copper. The difference is small, and it shows here that it's not significant in terms of milligrams per plant. Now, the results and discussion of tomato, for the tomato results. These first, we look at the dates, each dates that we harvested. So each one of these is a date that we harvested. So in the first day that we harvested in 85 days after planting, we see some differences in their marketable, their total yield. The 100th is the one that we see the most interesting difference because it's in the extra large tomatoes. And that had an effect in the marketables and the total yield. And then in the last day, we saw some differences in the large per carbon source. So this is all per carbon source. And this is carbon source rate of application. So in terms of number of fruit per plant, the cumulative yield per treatment, this is the same way that we were looking at the lettuce results. But for the tomato results, there was a difference in the number of fruit per plant cumulatively in spent mushroom compost and wheat middling compared to the rest of the treatments for large tomatoes. And that resulted to be different, but then also carbon rates. The carbon rate of application had an effect on the significant number of fruit per plant. The carbon source, the full rate of carbon source without the poultry manure resulted to be higher cumulative yield. So now the fresh weight. So this is weight. We're looking at number of fruit, and now we're looking at weight of fruit, fresh weight. And the biggest difference that we see is also in the 100 days after planting harvest, in which the carbon source had an effect on the large fresh fruit weight. And on the last day, on the extra large, the carbon source rate of application was significantly different, which had an effect on marketable and then full yield. So carbon source rate in pounds per plant for extra large, as we saw more or less in the last slide, had an effect on carbon source rate, had an effect on the extra larges, which was significant. Okay. So this is a little easier to look at. This is marketable fruit yield, and these are the dates of harvest. And the one in which we can see the most difference was in the 100 days after planting, in which all but molasses and untreated control had higher marketable fruit yield. And then per carbon source rate of application, the more differences that we can see is towards the last harvest, in which we can see more of the full rate. The full rate had a bigger effect on marketable fruit yield. So conclusions. First, I'm going to talk about conclusions on ASD, then conclusions on lettuce, and then conclusions on tomato. The conclusions on ASD is that using agri-food byproducts for ASD in Pennsylvania during the fall is possible. All products that were tested may be suitable for applications as carbon sources for ASD, but with middling, we're both highly anaerobic, and it has other benefits. It's highly available throughout the year. It's in ready-to-apply form. It's in a powder, and it's non-perishable as long as it doesn't get wet. But there's analysis to yet to be obtained. The data that we're presenting is still preliminary. Soil sample analysis, like the organic acids that were generated in the soil, are still yet to be obtained. With regards to the lettuce crop, there's no apparent phytotoxicity. We only saw small differences of sodium and copper, and the sodium accumulation was significant by application rate. With regards to the tomato crop, there was a carbon source effect on the large fruit number, and there was a carbon source application rate effect on the number of marketable full and total yield, as well as there were higher extra large tomatoes cumulative yield per application rate. So the full application rate without the poultry manure had a higher cumulative yield of extra large tomatoes, which is it translates to better marketability of your product and more product in general. We saw no interaction between carbon source rate of application and carbon source type. And overall, we had a big harvest. We had a big harvest without any fertilizer. So that, other than being like good news, where we can apply ASD and have some residual fertility, it also tells us like we need to adjust the application rates with regards to like our soil and the crop that we're planting afterwards. Also, the soil out there at the farm at the Rosso E. Larson is a heavy texture soil that probably has a lot of nutrition as well, a lot of fertility. So we might also be seeing the effects of the soil. And as well with the tomato crop, there's analysis to be obtained, the quality assessment, and the mineral analysis of the tomato that was ground and dried and pounded. I want to thank the support that I have received from first my lab. Most of them are here. Thank you all. And also the financial support from the USDA, Agricultural Research Service, Area Project, and ASD, as well as the Department of Plant Science and the horticulture farm staff. And thank you all for being here. So any questions? Yeah, that's part of my thesis. We're looking at the cost so far. None of the carbon sources except wheat middling have a market cost, a market value. Wheat middling, the 50-pound bag the 50-pound bag can be $10, and then you can also get a tote for like around $300. Yeah, and their value fluctuates throughout the year because it's used for cattle feed. The price goes higher towards the winter. But the other carbon sources really have no market whatsoever. And I even asked the people that we have interviewed so far, like if we were to buy something for you, if we were to find that this has some value and somehow it has demand, what would it go for? That just like a number? Like, they were like, no, we're not gonna sell it. Yes, yes. Like some of the time, I think, especially with spent grain, they have people, because the production is throughout the year, but it's not big. Like the batches of beer are not like it's not a lot of production. It goes like first come, first serve. So that will be the limitation if you don't have a connection with a person. Carbon sources for ASD. My understanding, ASD is not widely applied here in Pennsylvania. We did a trial last year, or 2 years ago almost, and we used molasses at a higher carbon rate because it was later in the fall. But yeah, so far like there's not like a standard carbon source for Pennsylvania, to my understanding. Yeah, that's that's what I understand. Yeah, well, yeah, cover crops have also been tested here because cover cropping is already like an approach that people are using for soil-borne health. No, for like soil health in general. Cover cropping has been used before, and buckwheat has been found to be a good supplemental carbon source for anaerobic soil disinfestation. But by itself, the anaerobic conditions that it can elicit are not that strong, are not that low. So it will be researched as a supplemental carbon source, like to cheapen the costs, to be used together with molasses. a really good question. No, we used the water that we had available. Um, yeah, to my understanding, I don't know if there's any study that has looked at the quality of the water before application. Um, it's being used commercially here in the US for the production of strawberries in California. Um, um, I think strawberries as well in Florida. It's still being used in Japan and in the Netherlands, also in Spain, it's being used. I'm not sure about places more in the tropics. I think it's more temperate zones are using it so far. Um, but yeah, the research and the interest is really high. The commercial applications are maybe not as widespread, but they're there, like at least in the US here. There's actually like a commercial company that sells carbon sources in California. Um, I will send you the link to what their name is. But there are that it's a business already. Yeah, yeah, that's the premise of the study that we don't wait. Like for example, like molasses is relatively easily available, but it's not produced locally, and prices, like might be prohibitive. If we can find something that is cheap and it's produced locally, it would be ideal. Arthropods. No, I don't have much knowledge about that. I know that for yeah, the minimal amount of time. It can vary. Some people some studies have applied it as short as like less than 3 weeks. Some people like more than 50 days. But the recommended amount for it to remain anaerobic is at least for 15 days, and the standard is 21 days, according to the literature. Yeah, the what I would say to a grower is that yes, all of the ones that we have tested can be used for ASD. So it depends more or less in your area, the area that you're and the resources that you have. If you have a chipper, that's something that can like, change the structure of the edamame needs further processing. So yeah, depending on your context as a farmer, but the one that we see that is like more widely like more potential for wide application would be with middle. because of the high anaerobic conditions, and it's non-perishable. Yeah, there's a couple of studies. There's a study in 2016 that Francesco published, and to my understanding, it didn't find more N2O emissions from the experimental plots with ASD, comparing them to the chemical soil fumigation, so the synthetic chemical approach. Yeah, so there's a couple of studies, and they have not found definitive data on the production of N2O being more from ASD. Yeah, if they have more inorganic nitrogen being applied, if they Thank you all.